Hi, I'm Gloria Muday. I'm a faculty member in biology. My laboratory studies plants, and we're interested in signaling pathways that control development, both hormone signals that initiate responses and transcriptional responses, as well as signaling molecules like reactive oxygen species. And so my lab has a number of graduate students, each working on very different aspects of these signaling questions to try to understand them in model plants and in agricultural plants like tomatoes, and in roots and leaves and pollen as they have questions that are best asked in those tissues. In my laboratory at Wake Forest, we study a couple of different signaling pathways. And one of those are reactive oxygen species. So ROS are really cool signals. At the right levels, they're really important and positive signals for development. At high levels, they can be quite damaging. And so plants, because they live outside and have high levels of light and temperature fluctuations, make a lot of ROS. So plants have evolved mechanisms to deal with those high levels of ROS called antioxidants. And some of them are called flavonols. And I'm standing by this brightly colored poster because the predominant color of flavonols in their downstream products, called anthocyanins, is purple. And so this just shows you a little bit of the biochemical pathway, and these particular structures are those molecules. We're interested in how plants make those and how plants use those to keep ROS in check. And so they're made at high levels in lateral roots, shown by yellow, and ROS is kept low in those same lateral roots because of the high levels of reactive oxygen species. They also accumulate in guard cells to control gas exchange, and in pollen to keep levels of ROS low so that pollen can productively fertilize. And these are all questions we're interested in studying. Another question we're interested in my lab are the signaling networks that turn on expression of genes to allow development to change in response to hormones. In particular, we're interested in the hormone ethylene. Ethylene is a gas, and it's produced by plants and well known for its ability to ripen fruit, but it also controls the development of roots. And so this poster highlights the work of one graduate student, Andrea Harkey, who really looked at the transcriptional responses to this hormone. We looked at the transcriptional responses using transcriptomics over time. We've also looked at changes in transcriptomics in receptor mutants that we know affect development. And so there are ongoing projects to really think about how ethylene remodels roots, how it increases reactive oxygen species, and how it changes expression of genes, making new transcription factors that remodel the developmental program.
Hi, my name is Jordan Chapman, and I'm a PhD candidate in Gloria Muday's lab. My research has focused on plant root development and how that's impacted by altered levels of antioxidants and oxidant compounds. A major focus in our lab is the antioxidant pathway, the flavonoid biosynthetic pathway. And so what I have done with my research is investigated mutants that are within that flavonoid biosynthetic pathway and look at how there are different impacts on root development based on the levels of flavonols and levels of reactive oxygen species. So flavonols are antioxidants, so they scavenge reactive oxygen species. So the more flavonols, the less reactive oxygen species. So ultimately, we determined that the flavonol camphorol is responsible for altering lateral root development, and overall, quercetin is responsible for altering root hair development. And both of these flavonols work through a reactive oxygen species mechanism. So they have lower levels of flavonols, they have increased levels of reactive oxygen species, and we get increased levels of root hairs and lateral roots. And so the next step of my project was to investigate what that mechanism is that reactive oxygen species are using to alter the plant development. So reactive oxygen species are able to alter proteins by modifying a cysteine thiol into a sulfonic acid. So this will alter the structure, the function, or the activity of the protein. And so one of the goals of my project was to do a proteomic approach to identify proteins that have that modification, that redox modification that I just mentioned. And so then we'll take a look at those proteins, and we identify proteins that are involved in root hair development or lateral root development. And so once we isolated those proteins, so we've begun to look at how they impact both root hair growth and lateral root development.
My name is Anthony Post-Leon, and I'm a third-year graduate student in Dr. Gloria Muday's lab. And my project focuses on how plants respond to drought stress, and more specifically, how they balance reactive oxygen species in their stomata during this time. So one of the first things that occurs in response to drought is plants need to close their stomata. This is where they lose water and where transpiration occurs. And so it's not beneficial to lose water when you don't have a lot coming in. So a way to not enter a water deficit in response to drought is for plants to close the stomata on the underside of their leaves. And one of the ways that they do this is through the upregulation of derivatives of molecular oxygen called reactive oxygen species. And so my project deals with the evaluation of both the localization and the spatial information of the increase of those molecules. And we do that through the use of a host of different reactive oxygen species probes or biosensors. And so this is an example here of an open stomata that would not be drought stressed versus one that is drought stressed and then closed. So we look at sort of how these reactive oxygen species are localized in the cell, as well as how much, and sort of how all those tie together.
Hello, my name is Melina. I'm a second-year graduate student in the Muday lab at Wake Forest University. In the Muday lab, we actually work with a plant species called Arabidopsis thaliana. This plant just has a quick life cycle, which makes it a little bit convenient for scientists to work with. It also has the entire genome sequenced. That basically means that we know all the building blocks, or most of the building blocks, that go into making this plant what it is. So we actually work with a bunch of different mutant plants. This basically means that the genome or the DNA of the plant has been modified in some way. We actually utilize a bacterium called Agrobacterium tumefaciens, and in nature this bacterium actually causes infections in the plant, and it does so by inserting a piece of its DNA into the plant's DNA. So we actually take advantage of this ability, and that's how we basically make edits into the genes that we want, and it disrupts the function. And so we have these different mutant plants, and we're able to look at them under the microscope and basically see if they have some observable characteristic that might be different than the normal plant. And this is called phenotyping. So we do a lot of phenotyping in the Muday lab, which is why I'm on a microscope. And I'm currently studying a plant hormone called ethylene. So if you didn't know, just like us, plants also have hormones that are involved in regulating different processes from when they're seeds all the way until the end of their lives. So this hormone that I'm looking at is called ethylene, and it's a 2-carbon gaseous compound. It's actually involved in a number of different processes, but some of which you're probably familiar with, specifically fruit ripening and aging and flowering and stuff like that. So I'm actually looking at ethylene in the context of plant roots. So when we treat plants with the hormone, we actually use a precursor to the hormone called ACC because it's a little bit easier for us since it's not a gas. So when we treat with this precursor molecule, we actually get an induction in root hairs. So you can see that here. This is a control plant right here. So what you're looking at is this, I guess, longish structure. That's going to be the primary root. And then these smaller structures here, those are single cell projections called root hairs. So this is the control plant right here, right? This has no hormone added to it. And then when we do add ACC, you can see that there's a bit of an induction here, especially at the very tip of the root. And so that's currently what I'm focused on.
Hello, my name is David Lee. I'm a junior at Wake Forest University majoring in biochemistry and molecular biology. In here at the Muday lab, I work with Anthony, a grad student, and focusing on the research of reactive oxygen species' role in guard cell closure. And it is so great for me to have the opportunity to work in Wake downtown to get research experience as an undergrad. And also it's fantastic to see that grad students also have the opportunity to mentor younger kids like me.